So I'm from Oxitec, which is a small company based in Oxford and established to set up and develop genetic technology, first developed at Oxford University, and we've been going about 10 years. And so insects cause a lot of harm to agriculture, as you'll all be very familiar, and also through transmitting diseases or viruses or pathogens to plants and animals and also to humans. And so a lot of my examples are going to come from mosquitoes, and that's partly because we've done a lot of field work with field trials with this technology against mosquitoes, and also partly so that by picking on one agricultural pest I don't underrepresent somebody else's favorite pest, and mosquitoes are a little more neutral perhaps in this audience. Okay, but in terms of what sort of species range do we have, and I'll talk a little bit later about why we choose these particular ones. We have some mosquitoes. I'll talk particularly about dengue. Probably for this audience, the tephritid fruit flies. So in Australia, that's particularly Mediterranean fruit fly and Queensland fruit fly, but you have some other ones. And we have products for Mediterranean fruit fly, and olive fly, which is a relative, I don't think in Australia, but a relative of Q fly. And then some other pests, some of them in Australia, and some of them potential invasive problems here, but not yet arrived. Okay, so dengue, won't go into very much detail, but it's a major mosquito-borne infectious disease. There's no therapeutic drugs, there's no treatment drugs, there's no vaccine. Sleeping under a bed net, which works well for malaria, doesn't work well for this because the mosquito bites during the daytime. So really the only way to try to deal with dengue, and so the mainstay of dengue control around the world is to try to kill the mosquitoes. And so this is really the state of the art of dengue control. So you can either try to find the larvae in their little pools, but they breed in small pools of clean water. So if you think about it at your home after a rainstorm, and I'm glad to see it does sometimes rain in Australia, contrary to rumour, then how many little pools of water do you have? And it's hundreds. And so to control this mosquito, you would have to treat every one of those every week. And if you do it, if you're in a city, and it's a very urban disease, if you're in a city and you do it, but your neighbour doesn't, or someone else, just one person on your block doesn't, that'll produce enough mosquitoes to transmit disease for everyone. So you really can't get enough of them this way. And so the alternative is to try to kill the adults, which is basically filling the whole volume of the environment with a toxic aerosol, insecticidal aerosol, which is not especially environmentally friendly. And it's also not very effective, because it doesn't penetrate still air well, and these mosquitoes are mostly indoors in closets and bedrooms and so on. And even to the extent that you do kill some, they're rapidly replaced from the immature pools. So basically the motivation for this research originally was we have to be able to do better than this, and then the equivalent for some agricultural pests. And so what we did, this is mosquito reproductive biology. Boy meets girl, sings little songs to each other, and the normal way of things, you get lots of little baby mosquitoes, and so the population continues and expands. So what might we do about that? Well, what if the male carried a gene, a lethal gene? So the male has 2 copies of a lethal gene, mates a female, 1 copy is carried in the sperm to the egg, and that, because of something we've done to the genetics here, that means that the mosquitoes don't develop through to the next generation. And so then if we can get enough of these females to mate our modified, let's call them sterile males, modified males, rather than wild-type males, then the target population will decline, and if we can keep that up, collapse over time, and then that's control. So that's the idea. And we call this system RIDL. So in this RIDL system, what are the key features of it relative to other control methods? So if you think compared with a chemical, for example, well, one is that these males will go out and look for the females for you. Okay? So you imagine a pot of chemical will just sit there, whereas these males will fly out to their flight distance, which for these mosquitoes is about 100 meters. For tephritid fruit flies, maybe 300 to 500 meters. They will fly out and look for the females over that sort of radius. Furthermore, these males will only mate with females of the same species and not with anything else. So the method is exquisitely species-specific. Now that's a feature of the method that is an advantage and a disadvantage in different circumstances. And so it helps point to what is a good target and a bad target. So they'll mate with females of the same species and not with other insects that might be in the environment, beneficial or neutral or mythical or whatever. So the species-specific. But on the other hand, if you have a dozen different pests all simultaneously eating your crop, you might prefer something a bit more broad spectrum rather than a dozen different species-specific methods. So it's just a feature of the technology that is environmentally very attractive and has some practical implications for and against. So this all might sound very innovative, but it is not. It is based on, well, bits of it are, but it's, hence in emerging technologies, whatever. But it's based on the use of radiation-sterilized insects for exactly the purpose that I described, which has been going on for 50 years or more. And there are a number of programs. One of the best-known ones is actually against the New World screwworm. This is a livestock pest. So it lays its eggs in wounds, in animals, and indeed in humans. And the larvae hatch out and burrow their way into the animal and kill it, which is all very messy. So it was eradicated, so this major pest was eradicated from the United States, and then from Mexico, and then down through Central America by a rolling program of release of radiation-sterilized insects. And then South America remains infested, and reinvasion of the north is prevented by continuous release of sterile insects in a barrier zone here in Panama. So this program has been going for years. It's highly sustainable, highly cost-effective, and just to reiterate, it has eradicated a major pest insect from a continent. Now, these programs have also been done on much smaller scales. So I'll come back to that. I want to talk about scale. This is a factory in Guatemala, run largely by the US government, producing sterile male Mediterranean fruit fly. This factory produces about 2 billion sterile male medfly every week. That's about 20 tons a week of sterile male medflies. So again, how to do all this stuff on a really big scale has been worked out, and it can also be done on very small scales. And one of the advantages for us is that although the genetics of our technology is very innovative, all the operational use has already been figured out with these radiation programs. So we know how to do this on whatever scale you like, up to continental scales, how to use this technology. Okay, so if you imagine, how does it work? Well, this is at a genetic level. I'll just touch on a little molecular biology later, but this is sort of Mendelian genetics. So if this was radiation, you'd irradiate this male, and you would you'd induce dominant lethal mutations in the sperm. So when this male mates a female, the sperm fertilizes an egg, carries with it the dominant lethal mutations, and that kills the offspring. It leads to the death of the offspring. But one of the problems with radiation is that you can't just irradiate the sperm and not the rest of the insect. So in fact, your radiation hits every cell of the insect, and that damages every cell of the insect in the same way as it damages the sperm. And that tends to weaken the insects and make them less sexy, or whatever. Well, it's actually a real problem for humans. But it's very hard to tell a sexy male fruit fly or mosquito from a rather puny one. The females know, but it's pretty hard for humans to tell. But it's a pretty fair bet that if you just fried it with radiation, it isn't going to be quite as good as it was before. So how do we do this? How do we aim to replace that? Is to have this male carry 2 copies of an engineered rather than radiation-induced lethal gene, and then you have exactly the same inheritance. Mates the female, all the offspring inherit 1 copy of this lethal gene, and then that kills them. Now, the little flaw with this plan that you may have spotted is that if 1 copy is enough to kill them, how do we make lots of males that have 2 copies, and why aren't they dead? And dead males don't mate so well either. So, of course, we have to be able to turn it off. So we arrange that we can turn off the lethal system so we can rear large numbers of them, and then when we release the males, then they do this. Because this is genetic engineering, we can modify a number of attributes. One is the time of death, so for mosquitoes there are reasons to want to kill them later. Another really important one is to make the lethality female specific. So if you think about pest insects, it's the females that cause all the trouble. We're just talking about insects here. So for example, with mosquitoes, only female mosquitoes bite and males don't. So all of disease transmission, dengue, malaria with different kinds of mosquitoes is all transmitted through humans. Excuse me, it is through humans, but it's also through mosquitoes, female mosquitoes. And then if you think about tephritid fruit flies, the first damage to the fruit is the female laying her egg in. It's the overposition sting damage, and that's only females and not males, and so on. And more generally, the reproductive potential of the population is the females. And you can modify the number of males quite a bit, and it doesn't matter, because the females all still mate. So we kill these, and then these males survive, but have 1 copy of the lethal gene, and so half of their daughters will die, and that gives you some additional benefit. I wouldn't want to overemphasize the effect of that. The big benefit is actually in the previous generation. So you have this population growing in your lab, you're giving them, or factory, you're giving them the antidote, but the ones you're going to release, you just don't give them the antidote. Okay? And they have a female-specific lethal, so all the females die, and that gives you a male-only population to release. And that's hugely beneficial. Because it's, not only does it not have sterile females that might themselves cause damage, but it turns out that and it's been done on huge scale experiments with Mediterranean fruit fly, if you release lots of sterile males along with lots of sterile females, the sterile males hang around and court sterile females that you released alongside them, rather than going out and looking for the wild females as effectively as they would have done if you hadn't just given them all those distracting females alongside them. And so in these big field trials, it's been shown that just not having the females makes the males 3 to 5 times more effective in terms of suppressing the target population. Plus, you don't, as a little extra bonus, you don't have to pay to rear those females that won't do you any good anyhow. So that's a pretty big deal. So essentially, this method I'm talking about, RIDL, is a sort of supercharged SIT. So operationally, it will look very similar, and so how to use it is pretty well understood. It can be applied to a much wider range of insects than radiation-based SIT because of this radiation damage issue, which is a real hindrance for some species. And relative to conventional sterile insect technique, what are the big improvements? Well, there's this genetic sexing that I just talked about, this ability to make a male-only release population that for tephritid fruit flies would give you a 3 to 5-fold improvement in per male effectiveness and additionally reduce your production costs. No radiation, again, reducing the process cost of having to do the radiation. It allows you to get down to a much smaller scale because you don't need a huge, great irradiator, and additionally improves the quality of the insects, the effectiveness of the insects, because you haven't fried them with radiation. For these kind of methods, when your control agent is a modified version of the pest and your target is the pest, you need to be able to tell them apart, because you want to know what your control agent is doing, and hopefully it's happy and there's lots of them and so on, and you want to know what the pest is doing, and hopefully it's unhappy and there aren't too many of them and so on, and for that you need to be able to tell them apart. Okay, so one thing, as well as the lethal gene, the other thing that we engineer into our flies is a marker. So this is it's just a fluorescent protein. So this is olive fruit fly, actually another tephritid, a relative of Q-fly. Wild type and our transgenic, under normal light they look exactly the same, like you'd expect, but under a fluorescent microscope, they're completely different. We're expressing a red fluorescent protein, and so you can really easily tell the transgenic ones from the wild ones, and therefore on a trap or in the field or whatever, you can tell one type from the other. So you can tell your control agent from the pest. Okay, so going back to these species, why did we choose these ones? What makes a good target, what makes a bad target? Because some of you may have ideas about what would be a good target, and some of you will know of pest species here in Australia that I don't know about, and certainly not as well as you do. So it's a species-specific method. So you need to have a single dominant pest that you care about, where controlling that one species will give you some economic benefit. A species that is already controlled by cheap, effective current methods, why would you change? Why would you do something else? So it's typically going to be used for insects that are a problem despite current methods. And sometimes that's current methods that work perfectly well but are losing registration or something like that. This is kind of trivial, but it is a mating-based system. And so if you have an insect that can reproduce without sex, like some aphids can, for example, then that might not be such a good choice because the females can carry on and reproduce without ever worrying about the males. Not too many insects like that, but some. It's useful, although not essential, for the released sterile males to be pretty harmless. So male mosquitoes don't bite, don't cause any harm, and so on. Same thing for tephritid fruit flies. But there are some other biting insects like tsetse flies in Africa, where both the males and the females bite. And so then if you release the males, you have to think about whether the sort of transient damage of releasing some males that might bite people there's a trade-off between that and the benefit of long-term population control. And you may well decide that trade-off is worth it, but you just have to think about it. You've got to be able to rear them. You've got to be able to rear quite a lot of these insects, and that varies from one species to another. There are some malaria mosquitoes that nobody's ever reared in the lab, so that would be an additional research challenge to figure out how to rear them. So we tend to stick to ones where the rearing is pretty well understood, and that's certainly true of the tephritids and a bunch of moths, for example, and some beetles and so on, for example. Short generation time, that's not essential at all, it's just everything happened faster. So everything in genetics the time really comes down to generations. So we can make the strains faster, and you'll see an impact faster if there's a short generation time. So all of these things have a generation time of about a month. This is a little faster, more like 2 weeks, and this one also. And then Mexican fruit fly, more like 2 months. So not essential, but helpful, and just moves everything along a bit faster. What could you do with such a tool if you had it? Well, the obvious thing is suppression. The point of this sort of approach is a longer-term population suppression tool. So often used, for example, the pink bollworm program in the US did this for many, many years. Start reasonably early in the season when there aren't too many of the pests, put in some steriles, keep on doing that. And they weren't even trying to suppress they were just trying to stop the pest population increasing as fast as it normally does. They're just trying to keep it below, so that it was below an economic threshold at the end of the season. Okay? And it's very effective for that. And then the minimum scale in which you can do that relates to the mobility of the insect. So a lot of the insects that I'm talking about, the tephrites fly 300 to 500 meters, the mosquitoes, these particular species about 100 meter. So you can do it on this kind of scale, sort of 4 to 20 hectares. If you're acres people, it's about double that. So that's sort of 10 to whatever acres. Now you can also, so SIT also has a fantastic reputation, probably since the days of DDT, the best thing for elimination. And so I showed you for the New World screwworm and also for Mediterranean fruit fly, it's been eliminated from California and Florida and so on by this kind of method. Now for that, you need a larger area and you sort of need everybody involved. You can't have one guy doing it and the next one not and so on and expect to get elimination of an area. So you need a carefully managed and coordinated program. It's been done repeatedly and very successfully with sterile insect techniques. I say probably the most powerful elimination tool there is today, but that might not be the place to start. You might want to start with something a little smaller. Okay, so I'm going to talk a little bit about going to the field. We do a huge amount of laboratory characterization of our strains, and I say we build them using a synthetic biology approach of the sort of thing that Lars described. I'm not really going to go into those details, but it just lets us build it in a modularized, standardized form that lets us transfer it from one species to another rather efficiently. And then I'll talk more about field data. So this is the only bit of molecular biology in the whole thing. So if aficionados want to talk to me afterwards, I'd be delighted to talk more, but strangely, most people don't. Okay. So this just shows you something about the precision that we can do. So if we use a promoter, which is a bit of DNA that controls gene action, that is expressed in the female indirect flight muscle. These are the muscles that power flight. They're in the thorax of the insect. And then expressed through this protein that I'll come back to, an effector, and in this case, it's just a fluorescent protein. It doesn't do anything except glow. You can see that if you look in this female mosquito, we see very strong fluorescence in the thorax. So indeed, it's in the right tissue, the flight muscles, and it's in the female, but it's not in the male. So the male thorax is not glowing. So this is indeed female-specific and indirect flight muscle-specific like it was supposed to be. Now this protein stuck in the middle here, this is called tetracycline repressible transcriptional activator, which sounds complicated, but it does exactly what it says. Transcriptional activator means binds on a bit of DNA and activates transcription. In other words, it turns this on. Tetracycline repressible means that it's tetracycline repressible. We're not very imaginative at naming things in this molecular biology game. So if you give them tetracycline, that acts as an antidote. The protein binds tetracycline, and then it doesn't bind here. If it doesn't bind here, it doesn't drive expression of this. And so then the insects don't glow. So if you look here at these females, these were reared on tetracycline, so in the presence of the antidote, and now they're not glowing. So you can indeed. So going back to how to make a lethal gene, because it's going to kill the insect, we have to be able to turn it off. This is how we turn it off. Okay? Just give them the chemical in their larval diet, and it's all switched off. Okay. So if you could run the video. So these are males of such a strain, but now instead of having a fluorescent protein expressed in the flight muscles, it's something that will actually affect the function of the flight muscle. So these are males in this cage, and they sit around pretty happily and don't do very much until you poke them, which you might say is a male characteristic. Again, we're just talking about insects here. And then they buzz around pretty happily, and these are perfectly normal-looking Aedes aegypti mosquitoes. But if we look at the next video, this is females of the same strain. Okay, females of the same strain can't fly. They can run around. They can't fly because we've done something in their flight muscles that stops them flying. Expressed something in their flight muscles. Now they can run around and jump a bit because those are muscles in the legs, and that just goes back to the specificity that we can get by this. We can express it in one particular muscle and not another, and affect even a particular type of movement. And as I pointed out before, it's female specific. Okay, that's fine. Thank you. So flightless mosquitoes, of course, can't survive in the wild. They can't get away from the breeding sites. They can't fly out and bite people. They actually can't mate even in the laboratory, because when I said boy meets girl and they sing little songs to each other, they really do. And the female wing beat frequency is a primary recognition factor for the males in this species, so males actually ignore them. The males, as you saw, have normal flight ability, as would the females if they've been given the antidote as larvae, which means we can grow large numbers of them when we need to. So collaborators, and all the field work that we've done is all with collaborators, partly for independence, so that somebody else is doing it other than just our little company, partly because we don't have the capacity to do it, and partly because if we're working in somebody else's country, it goes a hell of a lot better if you work with their government or entities or so on. So this is at Colorado State University. They set up 6 large indoor cages, and into 3 of them, these black ones, they once a week introduced males of the type that I just showed you, and those populations were driven to extinction. So this is a log axis of female numbers, whereas the untreated cages carried on happily. So at least in a small indoor, quite big indoor experiment, you can drive these populations to extinction. And one of the features there is that it's isolated population, so because it's a cage, so it's got walls, you don't have mosquitoes flying in and out. And that'll come up again later. And just to show that we don't just work on mosquitoes, this is the equivalent kind of experiment on Mediterranean fruit fly and on olive fly. These are done in Crete and diamondback moth in another research station. And in each case, the treated cages are driven to extinction, and the untreated cages carry on happily enough. So we can transfer this sort of technology. This isn't flightless. This kills females rather earlier. Transfer this type of technology to insect pests pretty easily across a pretty wide species range. Okay, so I've talked about this so far as if it's a sort of standalone technology, a single sort of magic bullet that will solve all these problems, which it is not. I don't really believe in magic, let alone magic bullets. But also, although it can work as a standalone method, it's better integrated with other processes, with other control methods. And there are a number of reasons for that. One of them is to do with resistance management, which I don't really have time to go into, but I'd be happy to talk about afterwards. This is a very good resistance management tool for other things that you use in conjunction with it. But there's also a fundamental issue of population dynamics, which is like this. If you spray or something like that, you can get the first half or whatever of the pest pretty easily, but it gets harder and harder to get the last ones. Okay? And that's true of pretty much anything you think about. If you went out with a fly swatter, it would be the same thing. Pretty much any control measure you think about, you can get the first whatever, harder and harder to get the last ones. With the sterile insect technique, both the radiation-based version and our genetically improved version, it's the opposite. It's easier to get the last ones than it is to get the first ones. And the reason is, if you pay a certain amount of money and produce a certain number of steriles, who the females mate depends on the ratio of sterile males to wild males. If the population is large, there are a lot of wild males, a lot of wild fertile males, and the females will see a lot of them and not mate your steriles so much for a given amount of steriles. Whereas as the population declines, there are fewer and fewer wild males and the same number of steriles, and even a higher proportion of the wild females will mate your steriles. So it actually becomes more effective as the target population decreases, rather less effective. And so if you think about that, that means that a sort of optimized mix of strategies is almost always going to involve using something to suppress cheap or whatever you got to suppress the target population to some extent at the beginning, and then a segue into, or probably a simultaneous use of, this sort of technology, both to get the last ones and then also to keep them down. Because if the target population is effectively zero, or it's actually zero or very low, then again you can keep it down with a relatively small number of steriles, in other words, relatively cheaply. So very much recommend thinking about it in terms of integrated pest management for these kinds of reasons. Okay, so in terms of actually going out into the field, with the mosquitoes, we've been out in Grand Cayman, in the Cayman Islands in the Caribbean, Malaysia, and also Brazil. And I'll talk a little bit about two of these. But before you go out into the field with I guess, any new technology, but certainly if you want to go and release a few million genetically modified mosquitoes, there's a bit more to it than just having a technically effective product. And you have to talk to regulators and the people concerned and get their approval. So we do that. This is community engagement. This is from our project in Brazil, where, again, always working with local partners. This is with the University of Sao Paulo and a social company up in the north of Brazil. Talking to people at slightly more formal sort of city hall, local government meetings, a lot of TV and radio to get the message out, local and national. Communication is very sort of culturally contingent, so this is Carnival, which is a big thing in Brazil, and these people dressed up as mosquitoes to go out and spread the word, which worked really well. Wouldn't work so well in Oxford, I have to say, and I don't know what it would be like here. But it just goes back to I mean, another really important reason why we always work with local partners is that they understand the local population, what is the best means of communicating people, what do they want to know, what do they know already, all these kinds of things. And then more formal sort of written communications, and the result is this. Let me just show you the trial site. This is for our Brazilian trial, and point out that it's pretty well isolated for mosquitoes on two sides, but not at all on the third side, and that'll come up again later. But in terms of the community engagement and what do the people think, so at this point in the trial, we were asking people's permission, but particularly because we were using a trapping method that we've gone beyond now, but basically you go into people's houses and you vacuum clean for mosquitoes. So it's called aspiration. So it's a pretty intrusive monitoring method. And over a number of rounds of survey, blue were all the houses where we went and people approved of the project and let us go into their houses and look for mosquitoes. Red are all the houses where there was no adult present to ask. And I would have in a third color the houses where there was an adult and they said no, but there were none. Okay, so we had 100% approval in the trial site for this GM mosquito project. That's in Brazil. Brazil has a big dengue burden. You know, dengue is a big deal. People understand that. Oh, and the result is this. This is one of our staff releasing GM mosquitoes off the back of a pickup truck, and everybody running around happy and laughing about it. Hopefully laughing with us, possibly laughing at us, but hey, happy. Okay, so then if you go to Florida Keys, very little dengue there. They had 100 cases over a few years ago. So very little dengue, very different demographic, relatively sort of anti-institution sort of mindset there. And also quite a bit of anti-GM activity there, and we at this stage had not done any sort of very little what are we about kind of messaging. So in that much more negative sort of scenario, nonetheless, a very substantial majority approved relative to disapproved of the potential use of GE mosquitoes, our engineered mosquitoes, by the local mosquito control agency. And pretty strikingly, surprising to me, although I saw a survey from the chairman of the panel group that said something rather similar in Australia, a rather higher proportion of the people thought that genetically engineered mosquitoes were safe than thought that chemicals and insecticides were safe. And this is what the control agency is using at the moment. Okay, so that's the community side. On the regulatory side, of course, we have been out into the field in 3 countries with the mosquitoes, obviously with national approval from the national regulatory authorities in each. For agricultural pests, probably the most significant single thing was an environmental impact statement, so the sort of highest level environmental analysis in the United States for tephritic fruit flies and also a moth, pink bollworm, so fruit flies and a moth. And the conclusion of this is all public, it's up on the web, their conclusion was that the incorporation of genetically engineered insects into their control programs is the environmentally preferable option. So it's a pretty striking statement from the US regulatory authority following their high level, the most detailed actually, analysis. Okay, so when you go through all that and actually do get out into the field, what happens? So this is from our trial in the Cayman Islands now, and just this is the area into which we were releasing the mosquitoes. And just to point out that, like the Brazilian one that I pointed out, it's well isolated for mosquitoes on 2 sides and not isolated at all on the 3rd side, and that'll be significant. So this is a population, tropical country, driven by rainfall, so it's driven by rainfall rather than temperature. This is the untreated area in green, and this is the treated area in red. And you can see that we reduced the target population, in this case by about 80%. And if you were to say, and the reason we did it this way is to have 2 very, very similar areas close together, so if it rains in one, it rains in the other, and so on. These are only a few hundred meters apart, these sites. This mosquito only flies about 100 meters. And if you were to say, 80% suppression, well, that's lovely, why didn't you do better than that? Which would be pretty harsh of you, because this is a really, really good result for mosquitoes. But a big part of the reason is immigration. So I pointed out that it's not isolated at this area, so the mosquitoes can just fly across this border, and in that situation, of course, you'll never get to 0. A slightly more subtle point is that this mosquito can lay eggs that can survive dormant for some months, and so there will still have been some eggs laid back here that were still hatching out here. So again, you'll never get to 0 in such circumstances. All right, so this was the Cayman Islands, which is this one here, 82% reduction. And then we went to a totally different area, a high-density urban setting in northern Brazil. So different country, different target population, very different ecological setting. But I showed you the shape of the trial site, again isolated on 2 sites and not on the 3rd, and we get very, very similar reduction in that site in northern Brazil. So then we went to a village outside of that city, which has not complete isolation, but better isolation than a suburb, and that's here, Mandacaru. And then we saw 90-odd% reduction by these indices. If you look at total eggs laid, which is sort of more linear measure, again, the same site, 99% reduction. And then if you look by a totally different trapping method, so look at the count adults rather than overtraps, this is the less isolated urban setting, 85% reduction, and that village, 96% reduction in the pest insect, in this case, Aedes aegypti mosquitoes. So just to summarize, I think, to use this technology, for widespread use of this technology against mosquitoes or ag pests or whatever, it's got to work, but also it's got to have public and regulatory approval. And so I hope I've persuaded you, well, I've shown you and I hope I've persuaded you that periodic release of these engineered males did indeed suppress target wild populations in multiple ecological settings. Although I was advocating using this as part of an integrated program, in those trials there was no conventional control. And the reason we did it that way is so that there wouldn't be any confusion about whether the result we saw was due to one or other of those packages. So they only had our sterile males, and that was therefore the thing that suppressed the target population. Sterile insect methods, radiation-based or ours, are not sort of instant kill things. So the males mate the females. They don't kill those females. They lead to the death of the offspring. So you have sort of generation delay. It's a longer-term population suppression tool rather than a sort of immediate knockdown tool. And it may take a couple of generations to get to the full effect. So this insect has a 1-month generation, so that's exactly what we saw. And although I didn't show you, the target population recovered only very slowly after we finished release in the Cayman Islands, presumably because we'd knocked it down so far, it took a while to recover. Public approval, again, as I showed you, initial indications have been pretty positive at the trial sites that we've worked with, very favourable responses. That may vary from one country to another, and indeed over time, based on the culture and the previous experience of GMOs and so on. Most people will have experienced genetic technology in the context of genetically modified insulin, which is what basically all injecting diabetics use, and GM vaccines, but they may not be aware that those are genetically modified products. So really, this means GM food, which is the best known case. For regulatory approval, independent national regulatory authorities have approved field use in the Cayman Islands, Malaysia, and Brazil, and also in the United States for pink bollworm, a moth pest of cotton. And then we've also imported into multiple countries, and the regulators, as you might imagine, you might say you're taking it into a lab, but they want to think what would happen if it got out because they don't entirely trust scientists to keep it where they say they're going to. So there's, and many other countries have approved importing under those circumstances. So this also, all three of these things, all three of the key things look pretty good. And then just very briefly, if they were interested in using this sort of technology in Australia, what would be a sensible first step? I would have thought some sort of pilot trial, just like the sort of thing that I was describing for the mosquitoes, sort of 10, 20, whatever hectare region, depending on the pest. We have strains already for Mediterranean fruit fly, diamondback moth. And although an individual farmer could do this in cooperation with us, probably if some state or national body oversaw it, it would have more credibility and more impact on the rest of the country. That's it. Thank you.